Hi everyone, thank you for coming. This presentation is entitled Phytochrome Regulates Cellular Response Plasticity and the Basic Molecular Machinery of Leaf Development. My name is Andres Romanowski and I did this work together with James Furniss and Ejaz Hussain at Karen Jane Halliday's lab at University of Edinburgh in the United Kingdom. So together with Karen, we posed the question, how does light control leaf development? In essence, we wanted to know how a plant could go from this phenotype to this phenotype just because they lacked a functioning photoreceptor called Phytochrome B. This also happens when plants are subjected to shade, for example. So one of the things that we saw is that PhyB9 mutants exhibit a lower leaf blade area than wild types. You can see a quantification of this in this graph over here, where we see the wild type values over there and the phytochrome knockout over here. Yeah? So how does this happen? Well, it could happen due to a reduced cell division, a reduced cell expansion, or both. So to answer that question, we did some DIC microscopy of different sections of the leaf. You can see two examples here. And we quantified the cell size, cell density, and cell number. What we found out was that this reduction in leaf blade area was mainly due to a reduction in epidermal cell number, as you can see here, whereas the cell size remained exactly the same, no significant changes. However, if we use a Phytochrome B9 mutant, we do not know if the timing of the Phytochrome B signaling is important throughout leaf 3 development. So in order to answer that question, we used a trick from the photobiologist playbook, which is to turn off Phytochrome B using end-of-day infrared. If you can see here in both images over there, end-of-day infrared treatment does phenocopy the PhyB9 leaf blade. So armed with this tool, we could ask ourselves, is timing important? To answer that question, we designed this experimental schematic that you see here, where some of the plants were kept the whole time under white light conditions, some of them were treated with end-of-day infrared since early in leaf development, some of them midway through leaf development, and some of them at later points, and since day 18 or since day 26. So using this particular design, we could ask, when does light stop affecting cell division in the leaf blade, and also if there are any other effects over time. Is it just cell division, or can cell expansion be affected as well? Well, to cut the story short, we found that if end-of-day infrared is applied since early on in leaf development, this phenocopies the results that we saw with the Phytochrome B9 mutant. So we have a smaller leaf blade due to less cell division, and we found no changes in cell size. However, if we apply end-of-day infrared much later in leaf development, we still see a reduction in leaf blade. But now, instead of affecting cell division, we're affecting cell size. So how does this happen? To answer that question, we designed an mRNA-seq experiment where we harvested plants that were subjected to end-of-day infrared since different moments in development, and we harvested early in development, midway in development, and late in development. Here we had to use, more than 150 plants per biological replicate, because as you can see, the tissue here for leaf 3 is very, very small. So in order to obtain enough mRNA, we had to harvest lots of plants. Then here in midway in development, you can already see the effect of end-of-day infrared over leaf 3. Here the leaf is much larger than here in the early untreated plants, and here you can see the gradual change that is due to end-of-day infrared being applied much later or much earlier in leaf development. So we did all the bioinformatics pipeline ourselves in our lab. We started with the raw FASTQ files, and we ended with gene expression analysis and also biological process analysis through GOTERM and KEK pathway analysis. One of the most striking things that we found was that the end-of-day infrared treatment led to more genes being downregulated than upregulated. And this also translated to the biological processes that we looked at. For example, downregulated processes were an order of magnitude 10 times higher in number than those that were upregulated. And I want to emphasize this because normally we look at genes that are overexpressed and we overlook those that are downregulated. But in the case of end-of-day FR, it seems that downregulation is more important than upregulation. So here on the right, we can see some bubble plots of processes that are being upregulated. Of course, we see the usual suspects: shade avoidance, response to brasinosteroid, ethylene signaling, response to auxin. And in the x-axis, what we see is the time component. So we see early development of plants treated since early leaf development, and this last point corresponds to plants that were treated late in leaf development. So these usual suspects were a way to show that our design was effective and was valid. And we could see that in addition to these classical responders, there are also others that are, for example, dependent of developmental time, such as, for example, flowering or histone demethylation or fatty acid beta oxidation. Now, when we looked at the downregulated ones, we could also see that some of them were affected all the time, such as DNA replication, and others were only affected, for example, early on, like leaf morphogenesis or cell division. When we took a closer look at this, we found that it was a concerted action that drives the reduction in the number of cells due to early on end-of-day FR treatment. We saw that basic cellular processes such as cell cycle, cytokinesis, DNA repair, and DNA replication were affected early on in leaf development. Here we can see genes that belong to the cell cycle, and here we can see the log-fold change of genes involved in DNA replication or mismatch repair. In addition to these processes, we also found some surprises. For example, we discovered that later in development, the plant affects, so the end-of-day FR treatment in plants affects translation processes and ribosome biogenesis. So this will lead to novel avenues of research involving light signaling and translation. So to sum up, end-of-day FR since early in leaf 3 development affects plate size, mainly to a reduction in cell number. But later in development, it still affects plate size, but now due to reduced cell size. We also found out, using a bioinformatics pipeline that we developed, that processes are affected in a time-dependent manner, such as the ones that you can see here. For example, cell cycle is only affected early and mid in development, while other processes such as, for example, translation, ribosome biogenesis are only affected late in development. This work has been published already in Plant Physiology, and you can find the whole story there. In addition to that, I developed a Shiny app where you can access through this link, where you can look at your favorite gene and you can make plots of log-fold change or normalized counts in all the different conditions that we use in our work. So white light, early treatment, or late treatment. Finally, I'd like to point out that our article was featured in Garnet, in Plante, and also in Plant Phys News and Views. If you have any questions, please feel free to forward them using my email or looking me up in my Twitter account. Thank you very much for listening.